Yes, everybody, a very warm welcome to the session, right? I hope that all your preparations are going up high, and today we are going to test your knowledge on the MCQs. This is Dr. Saranya. I am talking to you on a most important session. Yes, so this is a gist of important MCQs that you should definitely know. Yes, Kajal, very good evening. Right, happy everybody. So this is little bit about me. You might be familiar with me, but still I just want to introduce myself. I'm Dr. Saranya. I have completed my Ph.D. in biomaterials after 13 years of research and teaching experience. And this is my profile, right? You can find me at ResearchGate, and I am associated with Unacademy for a long time. Yes, so I am helping out in teaching off with your CSIR net papers. So all these are of high importance for your preparation, right? So let us begin our first question for the day. Yes, so one of your life history triad, right? So that is not characteristic of very small sized organism. Got it? So one life history triad. So that triad, yes, Rekapati, very good evening. So it is not a characteristic of very small sized organism. What it is called as, I mean, it is not a characteristic. Whether it is the delayed age at first reproduction, or it is going to be the earlier age at the first reproduction, or is it a high population growth rate, or it is going to be a short lifespan. Which of these is not a characteristics? Yes, Simran. So it is not a characteristics of very small sized organism. So this question will be easy if you know about your R and K selection. Right? Okay, fine, good. Yes, Simran. What about others? Come on. So the correct answer is going to be absolutely. It is the option 1. Exactly. Why option 1? It is because, right? So if it is going to be an R selected population, how will be the reproduction? It will take place at the early age. Yes, and there is going to be a high population rate and short lifespan. So what is the good example of R selection? Cockroach, isn't it? Right, so it yields a number of offspring, absolutely, and it is happening at early age reproduction. So where this delayed age reproduction is noted, it is noted only the K. Yes, Simran. So it is noted only with the K selection. Think of human. We reproduce at a later stage, not immediately after the birth. Got it? Yeah, so this is not a characteristics of the small sized organism. Yes, everybody else in this session, you are able to get this answer and justification, right? Okay, good. Very good, everyone. Keep it up. So let us make a move towards the next question. This is another question that we are discussing for the day. This is from your ecology. Alright, so shall we see this question? Right. So which of the following statement? Okay, so it is going to be most appropriate example of character displacement. Right, so you could have read this key term, character displacement. What is this key term, character displacement? Bring out the scenario in your mind, then definitely you will be able to get it off. Yes, okay, so just bring it in your mind, like what is going to be the character displacement. You will be able to answer this. Which of the following statement is the most appropriate example of the character displacement? Number 1 states that two related species depending on the same prey species, they avoid the competition. How? By feeding at different times of the day. Got it? Okay. The next one says that the body size of two related species, they are very similar when they are allopathic, but in geographical areas of sympatry, one species is smaller than the other. Right. The next one says that the food niche of the species is wider in the absence of the competing species than in the presence. closely related species, they can coexist if their densities are regulated by predator. So which is going to be the correct answer for this question? You can think and give me your answers. I'll give you a minute time. Yes? Okay, very simple question, but still you have to understand the question. Yes. So maybe the statements are true also, but you have to correlate it. Yes? Right. Yes. Good, everyone. Come on. Ready? Yes? All right. Come on, everyone. Absolutely. Right. Okay. So shall we see the answer? What is going to be the correct answer? Yes, everyone. Come on, you can give up your answers. If not, you have not yet given. Right. That's it. Yes, Simran. Yes, Supriya. Come on. Musani. Yes. Okay. Good, Raghupati. Right. So what happened? What is your character displacement? Character displacement is, yes, no, you are going to change the character. What is the best character displacement? You have seen the beak. Yes, the beak shape, it is changing depending upon the need. Absolutely. So what is the answer? It is number 2. Yes, Simran and Muzani, you got it right. Yes, Abhijit. So it is absolutely your option number 2. Yes. So what happens? The body size of the related species, yes, they are very similar. They are allopatric. Allopatric means they are in other region. But if they are sympatric, they are coming to the same region, then what will happen? One, it is going to be smaller than the other, then only it can it can survive, or else it cannot exist, it cannot coexist, right? So you can eliminate the other option. See here, the two related species, depending on the same prey species, they avoid the competition by a different time. If they eat at different time also, the food is going to get exhausted, isn't it? So they cannot coexist, right? So it is not a character displacement, right? The body sizes, they are very similar, but they are smaller than the other. Absolutely, that is the correct answer that we told. The food niche of a species is wider in the absence of the competing species than in the present. Is this a character displacement? Not at all. This is what? Number 3 is marking what statement? The food niche of a species is wider in the absence of the competing species than in the present. So what is this? It is nothing but your fundamental niche and the realized niche. Okay, so if the competing organism is present, then it becomes the realized niche. If the competing organism is absent, that becomes the fundamental niche. So this is not the definition for the character displacement. Yes, the next one, the closely related species, they can coexist, right? So if their densities are regulated by predator, it is the answer. Never. Yes, it is not the answer for the character displacement. Got it, everyone? Shall we make a move towards the next question? It is a very simple and a very good question from your ecology. In a population growing logistically and it is approaching your carrying capacity, okay, right, the change in density n per unit time, that is your dn by dt, this is attaining the maximum when n is equal to what? Wonderful question. So these questions are like Laddu questions from part B. Okay, so 2 mark you instantaneously get from these questions. Yes. So what about this question? What could be the correct answer? So in a population that is growing logistically Yes, Ragupati, very good. So it is growing logistically, no? So it is absolutely, it will obtain the maxima at your, yes, Kajal, yes, Musani, yes, Ragupati. Good, it is at K by 2. Very frequent question it is. So at K by 2, exactly. Yeah, Supriya. So that is absolutely the K by 2 where it is going to reach the maximum. This is the most repeated question. Right. Alright, the next question. Evolutionarily, with which of the following could parental care in animals be associated? Okay, so when will be this parental care be associated? Okay, so whenever there is a smaller clutch size, whenever there is polygamy, whenever there is greater longevity, or whenever there is going to be your semi-parity. Yes, when will be this parental care? It is very good associated with. Hmm. Yes, Avjit, absolutely your answer is correct. For the last question, K by 2, it is the absolute correct answer. Yes, it is like your V max by 2, isn't it? Yeah, true. Right. So as far as this question is considered, which of the following could this parental care in animal be associated with? So is it with your smaller clutch size, or is it with the polygamy, or greater longevity, or the semi-parity? Which of the following is associated with the parental care? Yes, Musani? Hmm. That's it. No other options, everybody? Yes, Avjit. Good. Musawni, good. Come on. The correct answer is absolutely your option 1. Yes, Ragupati. Oh, Kajal, don't worry. See here. If polygamy is there, what do you mean by polygamy? Many, no? So the mate is many. So it is not associated with the parental care. Okay, if it is mating with many, the parental care will not be there. Gone. Yes, and also this greater longevity, it doesn't mean that it should have a good parental care. But if the clutch size is small, absolutely it want to protect the species. Yes, so it will be absolutely helping in making the animals to be safe. So that is why it is there. Alright, so the change in a triad with time. Okay, right. So the change in triad with time as a result of natural selection. Yes, so this change in triad with time as a result of your natural selection is shown above. So this type of natural selection is directional, disruptive, stabilizing, or random. Yes. So which could be the correct answer? Very simple question indeed. Yes, absolutely. Yeah, Musani. Good. Others, I hope that you got the question. See from here, the graph is moving like this. Yes, so what type of selection it is? Yes, Musani, good. Yes, Kajal, Ragupati, good. So that is going to become your stabilizing selection, no doubt. It is absolutely the stabilizing selection. Right. So, now if at all the graph is moving here, what could be the selection? See here, I've shaded here. What is this selection now? If it is moving, yes, Avijit, good. So if it is moving towards one direction, one extremity, if it is moving towards one extremity, it is a directional selection. Got it? Right. So if it is moving towards the two extremities, okay, So what do you call as? Yeah, this is a stabilizing selection. So if it is moving towards two extremities, yes, very good. Yes, yes. No. So this, if it is two extremities, then it has become the disruptive. Got it? Right? But for this, the correct answer is stabilizing for this graph. Okay, but the question I asked was towards the one end. So that becomes the directional. This is the disruptive that has the two extremity. Yes. Got it, everybody? I hope that now it is clear. Yes. Okay. That's great. PCR based DNA amplification. Okay, so this is an essential feature of which of the following combination of molecular markers? Yes, Pooja. So if it is two extremities, absolutely it is going to be destructive. Good, Pooja Singh. Yeah, so the PCR based, yes Raghupati, PCR based DNA amplification, it is an essential feature of which of the following combination of molecular markers? RFLP, AFLP, SSR. AFLP, SSR, RAPD, RFLP, RAPD, SSR, RAPD, RFLP and SSR. Yes, all. Think and give me the answer. It is no hurry only. Yes, for your exam as well, you can take maximum 2 minutes for a question. Yes, Musani. Avijit, think. This marker always 1-2 mark will come. Okay, now then fine. If you're wrong also, it is okay. You can learn it. The correct answer is going to be 2. I'll tell you why it is 2. They asked that the experiment should contain the PCR based amplification. Yes. So if you take all these techniques, RFLP is restriction fragment length polymorphism. Yes, Raghu. So what is that RFLP? You take the DNA, you digest them based upon the restriction enzyme site. Okay, so your DNA will give various band. Now you can use the probe. So the probe will bind and it is going to highlight the bands that is bounded. Now you compare it with the parent or offspring or whatever it is you are going to compare. Yes, so now this is a parent A, this is a parent B. Okay, so this is a child C. Alright. So this is the mother, this is the father. Markers, they are made up of your sequence. Yeah. So they are the, yes, Avijit, don't worry, I'll tell you. So this is going to be, so we'll just see them in order such that you won't have any confusions here on, right? Number 1, it is going to be the RFLP marker. So this RFLP marker, it is based on the principle of hybridization. Okay, so it is a hybridization marker. Okay, so what is the difference between the PCR marker and hybridization? In PCR marker, you use the PCR experiment. In a hybridization marker, you don't use any kind of PCR method. You use only the hybridization technique. So in RFLP, what you do? You take the DNA, you digest them with the restriction enzyme. After digestion, you run them on the gel. So you get a variety of bands. Now after getting the band, you do this blotting and you have to convert all the bands from the gel to your blotting paper. Right. So after that, yes, you are going to add the probes. Okay. So once you add the probes will bind and you will get definite bands. Right, so they follow like your Mendelian inheritance. Okay, so they follow like your normal inheritance, allelic inheritance they follow. Okay, so like this is the father, this is the mother. So how will be the daughter? It will have one from each. Okay, so this is exactly, see, having this band and this is having this band. So this is the daughter of this one. Okay, so or the son of this one. Now you see one more thing you are having a band here. Okay, this is the D2. Is the D2 the daughter of F and M? No. Why? It has not got one band from each. This band length is totally different. Okay, so this RFLP, it is a hybridization markers. Yeah, the probes, they are radioactively labeled. Instead, you can use the fluorescent label also. Right? Okay, so the next technique is going to be this AFLP. So what is this AFLP? This AFLP, it is a very important technique. So this is the amplified fragment length polymorphism. So the mother of AFLP is going to be this RAPD and RFLP. Both put together you got this AFLP. So first we will know what is this. We know RFLP. Now we will know what is RAPD. RAPD is randomly amplified polymorphic DNA. Okay, so you have the word amplified. So do they use the PCR or not? Yes. Do they use PCR or not for RAPD? Yes, it is amplified, no? So they use the PCR. Yes, Deepak, happy to meet you back. So they use the PCR. So it is coming under the PCR technique. Yes, Avdit. Right. Yes, Ragupati. The next one, it is going to be your AFLP. Okay, so this AFLP, they use 2 PCR. Okay, not 1 PCR. They use 2 PCR along with your RFLP. Okay, so that is what it is, the AFLP. Got it? Right. Now you look at the question. SSR is absolutely Pooja. SSR is simple sequence repeat. Okay, right. Fine. So now you see this RFLP, you don't use PCR. Here also RFLP, here also RFLP. So only available answer is AFLP, SSR and RAPD. So apart from your AFLP and RAPD, SSR they use the PCR, SNP they use the PCR, right? So these are the different techniques that absolutely use the PCR for finding the molecular probes. Got it everyone? I hope that now this is all clear to you. Alright, good. So which of the following processes does not take place in 5' to 3' direction? Okay, yes, good Ragupati. So which of the following processes does not take place in 5' to 3' direction? Does not, that is the keyword. Yes, DNA replication, transcription, NIC translation, RNA editing. Which of them does not take place in 5' to 3'? Eliminate those which you know for sure that it is 5' to 3'. Then you will get on with which it is not there. Yes. So two things you will be definitely for sure that whether if it is going to come under 5' to 3' or not. Because the enzymes are meant with 5' to 3'. So eliminate that option and then look at it is going to become very easy for you. Yes? Okay. So what about DNA replication? Your DNA polymerase. DNA polymerase, it moves in which direction? 5' to 3'. So you eliminate this. The next one is transcription. So your RNA polymerase, it is also from your 5' to 3'. Yes, absolutely. So that is also eliminated. They ask which does not. What about NIC translation? NIC translation, where do you use and where do you use? Yes, that's okay. So where do you use this RNA editing? So which of them may be in the 5', may not be in the 5' to 3' direction? It is going to be your RNA editing. Why? After your RNA is edited, it goes back from 3' to 5' direction. It is an editor, no? So it reads in the reverse direction. Yes, you could have heard about your 3' to 5' exonuclease activity. Remember? Likewise, your RNA editing is also going from 3' to 5' direction. NIC translation is nothing but, yes, for NIC translation, yes everybody, for NIC translation what do? Where you could have studied this NIC translation, maybe in your DNA's footprinting you could have studied. Yes, okay. So in this Nick translation, what happens? They are going to create a single standard no break, right? So that single standard break is what is called as your Nick translation. Maybe you could have used the probe for that. Yes, right. So this Nick translation, you use your DNA pol only. Again, this DNA pol one, it will move in which direction? They will move only in your 5 prime to 3 prime direction. Got the point, everybody? Absolutely. I hope that it is now clear. So all these are gone and only RNA editing, your editosome complex and RNA editing, so that is only coming under your 3 prime to 5 prime activity, like your exonuclease activity. Yes, everybody, that's great. Good. So the deletion of the leader sequence. Yes, Sid. So the deletion of the leader sequence of tripoperon in E. coli would result in. Okay, so if at all you are deleting the leader sequence of your tripoperon, what will happen? It will result in decreased transcription of the trip gene. It will result in the increased transcription of the trip gene. Yes, Priya Kumari. And it is does not affect the transcription. It is decreased transcription of tripoperon in the presence of tryptophan. What will happen if you delete the leader sequence of the tryptophan? Yes, everybody, you are ready? Let us see the answer. Yes, all. I hope that you are all getting ready now. All right. Yes, all. Come on, I am waiting for you. Yes. So shall we see the answer, guys, if you are not answering? The correct answer is going to be your option 2. Why 2? See, this leader sequence is helping in what? It is helping in attenuation, isn't it? Right. So now you are deleting this leader sequence. Yes. So if at all you are deleting this leader sequence, what it will do? It will lead to the increase in the tripoperon. Do you remember how did we use this 1, 2, 3, 4? Yes, absolutely. So if there is going to be a pairing between 1 and 2, what will happen? 3 and 4 will pair. Okay, if 1 and 2 is not able to pair, then 2 and 3 will pair. Okay, so absolutely this is going to lead to, if at all it is deleted, it is not leading to attenuation. So it is leading to a continuation. So there is an increased transcription of the tripoperon. Yes. What is attenuation? Attenuation is nothing but it is the termination. Okay, so your RNA polymerase is not able to move. So it is leading the termination, but now this is deleted, so the termination is not happening, it is continuing to move. Yes, absolutely. So this blocking is not at all there, so it is happily moving through. So if it is moving through, there is an increased transcription. Got the point, everybody? So this leader peptide, it is responsible for all these things. So now you are deleting him, you are going to increase the rate of synthesis, or it is increasing the transcription. Yes, Regupati, I'll explain you one more time. See how this control is being made in your tripoperon. Okay, so imagine that this is going to be your leader peptide sequence. Okay, so this is the sequence, I mean it is in the tail. Okay, so this is going to be 1, this is the chain number 2, this is chain 3 and chain 4. Okay, so if at all your tryptophan is very low in the media, what will happen? Your RNA polymerase is going to move. Okay, right. So this RNA polymerase is moving and your tryptophan gene, your tryptophan mRNA is present here. Okay, so what will happen? Your RNA polymerase is going to move through this region, right? So now there is no termination. All right, fine. So if at all there is going to be too much of tryptophan in the media, Okay, so if there is going to be too much of tryptophan in the media, what will happen? Your RNA polymerase will stall here in the tryptophan because it is not needed. No, too much high it is there. So what will happen? This one will not be available to pair with the 2. So this has a complement sequence. So 2 will pair with 3. Okay, and now 4 is free. So this is leading to the termination. So when it will terminate? Whenever there is a very high tryptophan. Imagine the reverse condition. Your tryptophan is low in the media. Whenever the tryptophan is low in the media, what will happen? Yes, whenever your tryptophan is low in the media, this RNA polymerase is moving here. So whenever the RNA polymerase is moving here, your 1 and 2 will pair. Likewise, your 3 and 4 will pair. So it is not leading to any stoppage. It can very easily move. So if it is moving, your transcription is on. Okay, right. So who is responsible for this attenuation or for this termination? It is going to be the base pair between 2 and 3 of the leader peptide. Now this leader peptide only you are, nice, thank you. Yes, so now this leader peptide only it is now responsible for that. Now you are deleting this leader peptide. So your tryptophan synthesis is on always. Got it everybody? The logic behind this question? Yes? Good. That's great everybody. Come on. So let us make a move towards the next question. It is a very direct question from your bacterial genetics or your microbial physiology. Yes? Okay. Fine. So this is an important 4 mark. Right. So see this question. A bacterial culture that is undergoing balanced growth. Okay, yes, so it is undergoing a balanced growth. So this is requiring a first order reaction. In other words, the rate of increase in a bacteria at any particular time, the rate of increase is proportional to the number or mass of the bacteria that is present in the time. Right. So they have given that if n is the number of cell at a present time t, mu it is the growth rate constant. Okay. So what is going to be your dn by dt? Very direct question it is. Still you can able to give a try for this question. What is going to be your dn by dt? It is the change, isn't it? Yes. So change. So let it be dn by dt or let it be your dt by dn also. You are going to find out the correct one. Yes. I am not going to tell the answer. You are going to find it out and let me know. Very direct formula. So we have learnt a number of formulas for that. Yes. So your dn by dt it is equal to R max n we have learnt. Yes. Remember that? Yes, Masavni. And then we have learnt that your nt it is equal to n naught into 2 power n we have learnt, and your n is equal to log of nt minus log of n0 by 0.301 we have learnt. Yes, and your k it is equal to n by t we have learnt, and your g it is equal to 1 by k, so that is equal to t by n we have learnt, and then your td it is equal to log n2 by r we have learnt. Good everybody, come on. These are the important formulas that we have learnt. Yeah, so no doubt the correct answer is absolutely going to be 1. Yes, Musoni, Raghupati, Kajal and Avjit. Very good. So your dn by dt is equal to mu into n. So n is the number, mu is the specific growth rate. Instead of R max, here we are using the mu. That's all. Got it everybody? I hope that it became very simple for 4 marks, right? Yes, very simply you get 4 marks for this question. Right, good everyone, keep it up. Okay, so see this question, very interesting question. A typical animal cell that is nucleated. Okay, so you have a typical animal cell. Okay, yes, oh sure. Yes, Syed, I'll definitely take a subject wise class. Noted. Yes. So this is a typical animal cell. So that is going to be nucleated. All right. So this membrane, it is going to contain, yes, glycolipids and glycoproteins where in the plasma membrane. So this is containing your glycolipid as well as glycoprotein where in the plasma membrane. Right. So to determine its topological distribution, lectin, it is used as a probe. Okay. Right. So the following interaction may be the basis for the probing method. So you will study this protein-protein interaction, protein-sugar interaction. What is this lectin? Just think. Okay. Right. Where the lectin is going to interact with. That is what in affinity chromatography could have studied this. Okay. So protein-sugar interaction, protein-lipid interaction, protein-sterol interaction. So which could be the correct answer? Yes. Good. 4 marks. Correct, Avjit. Absolutely, you are right. So this lectin, it is a sugar binding protein, isn't it? Correct. So you will be able to absolutely find with yes, no, it is a sugar binding one, right? Yes, Kajal, yes, Supriya. So it is a sugar binding one. So your lectin will bind only with the glycoprotein, right? So this is a glycoprotein, yes, and it is going to bind with your lectin. So this is going to be a sugar and protein interaction. Okay, so only your answer B is correct. So protein-protein interaction, lectin is not a protein. Protein-lipid interaction, this is also not true. Glyco is not a thing. And then protein-sterol, it is not true. So only available answer that is true is your protein-sugar interaction. Yes, everyone, clear? Okay, the next. During many important cell processes, many proteins, they need to undergo degradation. Why? To culminate a part of the process. For example, during the cell cycle, cyclin proteins, they need to degrade and they allow the cells to exit the mitosis. Okay, so who is helping in your mitotic exit? Yes, so you want to do a mitotic exit. Yes, this is achieved by selective ubiquitinization of cyclin, right, followed by its degradation by the proteasomes. The specific protein factor that is involved in this process, it is called as the anaphase promoting complex APC. Okay, so what kind of protein or what kind of enzyme it is? It is an E1 enzyme, E2 enzyme, E3 enzyme, or it is a protease. So you should remember this about your ubiquitin and then click the option, isn't it? Yes. So which one is going to be the correct answer? Yes, all. Got it? So we have studied about the APC, no? So APC is what? Huh? No. This is degraded by the proteasomes. So the first option is to remove the proteasomes. Yes, Shika. Proteasomes cannot be the answer. Why? Proteasomes are only degrading them. Right? Yes. So who is tagging them? Think of. It is none other than your E3 ubiquitin ligases. Yes. So these are the E3 ubiquitin ligases. We could have seen this in your APC complex, APC bar C, right? Yes, Shika. So they are the E3 ubiquitin ligases. So what they do to the cyclin, they add the ubiquitin tag. Once this ubiquitin tag is being added, then the proteosomes, they recognize this tag and then they will degrade. So that is why you first eliminate the proteases. These are the enzymes that help in this process. E2, it is also there to help it, right? But the actual specific protein that is involved in your anaphase promotion complex is E3 ubiquitin ligases. Very important question, guys. It's okay if you have not yet studied also, it is okay. Please make sure that you learn this concept. And for the G1 phase, which one is coming? It is the SCF. Okay, so that is also adding a ubiquitin tag. SKP, Qlin, and then F box protein. So they also have the association of the E3 to add a ubiquitin tag to them. So the G1 cyclin will be degraded. Got it, everybody? Very important question it is. Okay, this question may be pretty long for you. Yes, it's okay, Kirti. Yes, Shika. Yes, Rekupati, Supriya, Kajal, Sid, Abhijit. Yes, everybody. Who else there? Musani. Right. Good, everybody. Keep it up. It's okay. Let it be right or wrong. That's okay. Yes, Priya Kumari. Right, everyone. Good. Come on. Yes. So this is a pretty long question to carry out, but still it is very, very easy question. Yes. Okay. Fine. Very long, right? I'll read the important part only. The 3' to 5' exonuclease activity is not associated with all the polymerase because it is present only with, yes, your 3' to 5' exonuclease activity. Yes, it is present only in, with whom it is present. Very important, guys. So where it is present? Yes, Shika? 3' to 5' exonuclease. Your E. coli DNA polymerases, but not all the mammalian polymerases. What about Pol 1, Pol 2, Pol 3, Pol alpha, Pol beta? Yes. Who contain it and who doesn't contain it? Yeah. So shall we see the answer? We are almost reaching time. The correct answer is going to be your option 3 with your A and C. I'll just tell you what it is. Okay. So if you take your DNA polymerase, they may contain your 5' to 3' exonuclease or 3' to 5' exonuclease. Only your Pol 1 has your this fellow, but everybody is going to have, yes, everybody is going to have your 3' to 5' exonuclease activity, right? Yes. So what about your yes, so what about your this option? What about your Pol A? Will your Pol A have it? Will your Pol B have it? Yes, Musavni. Right? So what about your Pol alpha and Pol beta? Pol beta, it is associated with what kind of activity? It is associated with a repair, right? Pol alpha, it is associated with primase. Yes? Okay. Then what about your D and E? Definitely they are into your lagging stand and the leading stand, right? Absolutely, yes. So this 3' to 5' exonuclease activity, it is not associated with, it is not associated with, but it is present only in this E. coli polymerases, but not all the mammalian polymerases. A is there always. In all the options, A is there. And no, with your C, it has your Pol 1. Definitely Pol 1 has your no exonuclease activity. So definitely you can include that option C, right? And no, so these two are going to be the correct answer. Yes? Okay, fine. So what about your rho factor? This rho factor, very long questions, no? As of your molecular biology is considered, but still no, this is pretty important, right? Okay, so no, what about your rho factor? this rho factor, it is being involved in termination of the transcription. So your rho dependent termination requires the specific recognition sequence, isn't it? Right. So if at all it is a recognition sequence is getting required, right? Okay. So it is, you know that there are two types of your rho, termination. One, it is going to be this rho dependent as well as the rho independent termination, isn't it? Right. Okay. So these are the rho dependent and the rho independent termination, and this genetic manipulations, they indicate that yes, the rho dependent termination, they require the recognition sequence. Okay, and so it will make of a move only in the new synthesized in the upstream side, and the recognition sequence, it must be on the nascent RNA rather than the DNA. Okay, so the full features are elucidated, and they were consisting of around 80 to 100 nucleotides. Okay, so which could be the correct answer, they are asking. Maybe it is pretty too heavy for your YouTube session, because I think that it is very difficult for you to view the question. Is it so? You will take this as a homework. You will just take the snapshot and you will try it as a homework. Yes, will you do that? Yes, everybody, I hope that it is pretty big for you to read now. Maybe you will take it as a home assignment and you will try it. Yes, everybody in this session, you will do this. But this is going to be, yes, Shika, absolutely correct. So this is going to be a very important thing for you, because in your exams also such big questions are coming. Yes, absolutely, answer 1 is right. Yes, so in your exam also you will get such big questions. You should have the patience to read the question and take off, right? Absolutely, the 1 is the correct answer, right? So this is another simple question that we can solve. Yes, so you are working with an in vitro eukaryotic system which produced both the capped and uncapped mRNA. Okay, so this is the capped fellow, this is the uncapped fellow. So you incubated these mRNA with mammalian cell nuclear extract and quantified them. Okay, right. So you see that the level of mRNA here with the capped, this pre-mRNA level is less. Here this pre-mRNA level is high. The spliced level is high. This polyadenylated level is high. Here these two are low. Okay, so which one is correct, they are asking. Okay, here you see that capped pre-mRNA, it is empty. Okay, this is going to be the spliced one, right? And this full black is going to be your, who is that? So this is the spliced one and this is the poly A tail. Got it? Yes, so which could be the correct answer? Yes, Avijit. Got it? Ready, everybody? Yes, Sid. Which is the correct answer? The correct answer is absolutely your option A. Why option A? See here. Yes, Musani. So during your RNA processing, what happens during the RNA processing? Yes. See, this is going to be your pre-mRNA. Yes, Shivani. So during this RNA processing, the pre-mRNA is going to added with the cap and then the tail. Okay, so this becomes the processed RNA. Got it? Right. So now what happens? This is the poly A tail, this is the cap. Okay, fine. So whenever it is capped, will there be a pre-mRNA? Not at all. Okay, so that is why the pre-mRNA level decreased. But if it is uncapped, it is unprocessed. So this pre-mRNA level is high. By that only you could have got it. Yes, Shivani, absolutely. Yes, right. Here what is wrong is that in uncapped, this spliced is more wrong. In uncapped, spliced is more wrong. Here in in capped they are telling that poly A is more and splicing is less. This is wrong. Why this is wrong? Because after splicing only you have the poly A. I mean after poly A only you have the splicing, right? Yes. So that is no, after capping only you can do that, or else it cannot happen. Yes, yes, absolutely. This is a most repeated question also. Yes, everybody, I hope that you try to solve a lot of questions today, so you can unveil still more practice question with detailed solutions. That is all the recorded versions are available. So for the new batch we will beginning up with, but for the current batch all these recorded sessions are available with complete set of the courses. If you really wish to continue learning with this, you can log in to Unacademy and you can create a login for you. So that creation of login is for free. You can attend a lot of special classes. If you seriously feel that it is worth of it, you can come up with a subscription. Yes, so at that time you can use my code SaranyaLive. See that S and L, it is capital. Yes, absolutely. So if you use this code, you will get a 10% off on the total fees, right? Yes, so absolutely you can do that. A lot number of things are still there for you, waiting for you. Yes, so I think the time is almost up. So I have to wind up with my presentation. All right, but you have upcoming no very important one. Yes, Abhijit. Oh yes, that's nice, Abhijit. Thank you. You can listen to the other special classes and other YouTube sessions also I do have at the freeware. So no, at Unacademy YouTube series only we have I have conducted all the important topics, right? So these markers, everything you can just have a quick view through that. Yes, applied biology, it is a booster unit. Yes, Shivani. So the next YouTube session no, it begins again from November 1st. Okay, and the special class I have put it on tomorrow. Yes, so at 7 p.m. there I am going to tell you the checklist topic that you should have prepared for the examination. Likewise from November 1st. Oh yes, sure, see. So my ID is biovsa16@gmail.com. Okay, you can reach me out for queries. Yes, Yashika. Definitely. So unit 12 is the applied biology. So that only it is part B is coming much. Yes, I know that unit 13 you could have prepared because part C it is very good as booster. Likewise for part B also it is a booster, but these years current years I find that unit 12 is a real time booster, right? So don't miss out to read out the important topics from that. So this is about this Revise India, where we have planned with many top educators along with me. They are going to give you a number of free classes for you. I am doing it unit wise for you. So my time slot is at 2 p.m. So join me in that and get benefited and get the edge over the preparation. Yes, everybody, I hope that you are all happy with our this thing. So this is the course fee for 3 months. It is around 5.2. You can give your ICMR examination, for your GATE examination. Apart from our CSIR net, you can do all in all one preparation by subscribing to this amount, and after discount it is going to be very less for you, right? So enroll at the earliest and get the edge over the preparation. Thank you guys. I will meet you soon in another interesting session. Bye bye. Take care till we meet. Thank you all. Any doubts, just reach out me. Yes. Bye bye all. Thanks a lot. Good. Have a good night. Yes, Sophia. So except me, who is teaching is Abait Hussain and Dr. Debashishkar. Okay, he is also teaching for English. So long time English batch was asked for. Yes, so no, we are having a batch course in English only. Okay, so in this course you will apart from me, Debashishkar and Abait Hussain, both of them they are teaching in English. Okay, so you need not worry, like no, earlier we didn't have much faculty on English, but now many faculties are there for English as well. Yes, Sophia, got it? Good, good. Any other queries, you can definitely reach me out. Yes, so you will get a full preparation, don't worry. Yes, okay. That's great. Yes, Sophia. Take care. Oh yes, sure, Sophia. See, apart from me, Dr. Debashishkar, okay, and Abait Hussain, okay, so they are also taking up in English. I told you. Yes, absolutely. Yeah, yes, Diksha, 7 units. Yeah, so for part C, 7 units are enough. Okay, but for part B, what you can do is that another 2 units you do a rough study. Okay, because part B it is going to be very essential, right? So you do a rough study for 2 more units, you can be very confidently attempting your part B. Okay, definitely you need a 9 unit strategy these days. Yes. Okay guys, that's good. No other doubts? Yes, Sophia. Okay, that's good. Everybody take care, all the very best. I'll catch you up in another interesting session. Thanks for joining. Have a good night. Yes, all.